Kia ora team, welcome back to the 2.5 Genetic Variation and Change 4-credit external video series. This is video 12. In this lesson, I'm going to introduce you to population genetics. You will learn about gene pools, genetic diversity, allele frequencies, and allele fixation. I'll also give you a quick overview of the processes that change allele frequencies. These are natural selection, mutation, gene flow, and genetic drift. By the end of this lesson, you should be able to define those key terms and calculate the allele frequencies in a gene pool. Let's get started. Before we dive into population genetics, let's quickly review some of the basics. Every trait or phenotype you see in living things is coded for by genes. Genes are stretches of DNA that code for proteins, and proteins build the traits we observe. Many genes come in different versions called alleles. For example, let's zoom in on this gene for hair texture in humans. We can see that there is a tiny difference between the DNA sequence of the hair gene at the top and the hair gene at the bottom. The hair gene at the top has a C nucleotide, while the hair gene at the bottom has a G nucleotide. And that tiny difference in the gene creates two different alleles, producing two different hair proteins that produce two different hair textures, curly and straight hair. Changes in DNA could lead to changes in traits. Genetic variation, or differences in the DNA code, can lead to phenotypic variation, which are the differences in the observable traits. Up until now, we've looked at individuals, families, or single crosses, but population genetics zooms out to look at the whole population of hundreds or even thousands of individuals, all carrying different alleles. This big picture view helps us understand how species change over time. Let's go over some key terms in population genetics. In biology, a population is a group of organisms of the same species, living in the same place, and is capable of interbreeding and producing fertile offspring. So population genetics is really just the study of how allele frequencies, the proportions of alleles, change in a population's gene pool over time. The gene pool is the complete set or the sum of all the alleles present in a population. And genetic diversity means the range of different alleles present in a gene pool. A population with lots of different alleles has high genetic diversity, which makes it more likely that some individuals can survive if the environment changes, whereas a population with low genetic diversity is more at risk. Let's put those terms into a New Zealand example. Here is a beautiful population of takahe found in the Fiordland alpine tussock grasslands. These takahe fit our definition of a population because they're a group of organisms from the same species, living in the same area, and can interbreed and produce fertile offspring. I'm going to make up a gene with two alleles, let's say gene A with the big B allele and the little a allele. The takahe gene pool here is made up of every single allele present in this population of 11 takahe. So this takahe gene pool has 22 alleles in total, 7 big A alleles and 15 little a alleles. In this gene pool, we can say that there is some genetic diversity because there are two alleles present in the gene pool. If all our takahe were homozygous recessive, then there would only be one allele present, the little a. And in that case, we would say there is no genetic diversity in the gene pool. In reality, a population with many different alleles has high genetic diversity, which makes it more likely that some individuals can survive if the environment changes. But if a population has low genetic diversity, it's at risk of extinction. Sadly, takahe have very low genetic diversity because their population once dropped to fewer than 100 individuals. That bottleneck event reduced their alleles, making them more vulnerable to disease and less able to adapt if the environment changes. You will learn about the bottleneck effect and genetic drift in a future lesson. By contrast, New Zealand sheep population has high genetic diversity. This is because selective breeding programs maintain lots of different alleles, which keeps the gene pool large and varied. When we talk about changes in the gene pool, we often use allele frequencies as a measure of this change. Allele frequency is the proportion of a particular allele in the gene pool, and it tells us how common or how rare a particular allele is in that gene pool. To calculate allele frequency, you have to take the number of copies of that allele, so the occurrence of that allele in the gene pool, and divide it by the number of alleles in the gene pool for that gene. Let's work through some examples of calculating allele frequencies. To calculate the allele frequency of the big A allele, we need to count how many times it appears in this gene pool. That's the occurrence. And then we need to divide it by the total number of alleles in this gene pool. So, how many big A alleles are there? 1, 2, 3, 4, 5, 6, 7. And we need to divide that by the total number of alleles, which is 22. We know that because there's 11 individuals. So 7 divided by 22 is 0.32, multiplied by 100 to turn it into a percentage, and that's 32%. To calculate the allele frequency of the small a allele, we need to count how many times it appears in this gene pool. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. That's 15 times. Then we divide it by the total number of alleles in this gene pool. That's 22. So 15 divided by 22 is 0.68, multiplied by 100, that's 68%. So the recessive little a allele has more than doubled the allele frequency of the big A allele in this gene pool. Here's another example. Calculate the allele frequencies in this population of P waka waka. To calculate the allele frequency of the big P allele, we need to count how many times it appears in the gene pool. 1, 2, 3, 4, 5, 6, 7. We then divide that by the total number of alleles in this gene pool. How many alleles in total? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 7 divided by 10 is 0.7 times 100, 70%. Then we need to calculate the allele frequency of the small p allele. So we need to count how many small p alleles there are, divide it by 10. So 1, 2, 3, 3 small p alleles divided by 10 alleles in total equals 30%. So as we can see in this case, the big P allele has more than doubled the frequency of the small p allele. Now it's your turn. Pause the video to calculate the allele frequencies of these 3 alleles: big A apostrophe, and little a. There are also 2 populations that you need to calculate for, population 1 and population 2. Make sure you show you're working, and I'll put up the answers in a few seconds, so pause the video now. Here are the answers. Let's look at population 1. The total number of alleles in population 1's gene pool is 46. There were 26 big A alleles, which means an allele frequency of 57%. There was 1 new allele that arose through a mutation, A apostrophe, which means an allele frequency of 2%. And there were 19 little a alleles, which means an allele frequency of 41%. Okay, now let's look at population 2. The total number of alleles in population 2's gene pool was 38. There were 11 big A alleles, which means an allele frequency of 29%. There were 27 little a alleles, which means an allele frequency of 71%. Hopefully you got those all right. So why do we need to calculate allele frequencies? We need to be able to calculate allele frequencies so that we can track changes to gene pools over time, measure genetic diversity, and compare populations to predict future changes. Allele frequencies are like population health checks. They tell us what alleles a population has, how common they are, and whether things are changing over time. There are a number of evolutionary processes that can affect allele frequencies and cause evolution of species. We're going to cover them in more detail in other videos, but here's a quick overview of them. In the evolutionary process of natural selection, some alleles give an individual an advantage in the ability to survive and reproduce. As a result, those advantageous alleles become more common in the gene pool. In the evolutionary process of mutations, brand new alleles can enter the gene pool through random DNA changes. That's what the apostrophe notation means. It's a mutation. If the mutation is advantageous, the new allele will become more common in the gene pool. In the evolutionary process of gene flow, individuals immigrating into a population or emigrating out of a population could bring in new alleles or take alleles away from the gene pool, causing allele frequency changes. And finally, in the evolutionary process of genetic drift, random chance events can change allele frequencies. The effect of this kind of change is a lot more dramatic in small populations. We will go over each of these 4 evolutionary processes in future videos. So as you can see, allele frequencies can change from one generation to the next, depending on the evolutionary processes acting on the population. Sometimes, an allele can spread across the gene pool until it's the only one left. This is called allele fixation, where the allele frequency reaches 100%. But most of the time, allele fixation is not good because it reduces genetic diversity. With fewer alleles in the population, the species has less flexibility to adapt if the environment changes. And once an allele is lost, it's gone for good unless it reappears through mutation or is reintroduced by migration. So that's all for this lesson. By now you should be able to describe the terms population, population genetics, gene pool, genetic diversity, allele frequency, and allele fixation. You should also be able to calculate allele frequencies in a gene pool. Thanks for watching, and I'll see you in the next video.